Hi, welcome to Chapter 29, Heredity. I wanted to show you this really cool multicolor karyotype. So basically they can fluorescently label each individual chromosome based on, like, they match, right? We have our A, T, C, G content on the chromosome, and then they can match a fluorescent tag with a specific color, and so you can see that the fluorescent dye will bind to each chromosome. And so multicolor, there's many colors, and we can see if there's any issues or problems, right? So this karyotype will be done in a lab. We have computer programs that sort them all out for you. Before, you'd have to sort them out by hand. Okay, these two are the same, right? So here's chromosome 1. So you can imagine it was kind of like solving a puzzle. Now we have nice computer programs that sort them all out for you. You can see this is a female, right? Because there's 2 X's here. So let's go through this heredity chapter. I'm not going to cover this in great detail on the test. I'm going to ask you basic questions, so stick to your learning objectives for this chapter. What is the definition of genetics? So it's the study of the mechanisms of heredity. How do we pass information on from one generation to the next? So we know that genes contain DNA, and this is going to be the recipe or the blueprint, for the synthesis of proteins. So hopefully we remember that DNA is transcribed into RNA, and then RNA is translated into a protein. So if I have this chromosome here and this chromosome here, let's say right here I have a gene, and that gene is for hair color, right? So it's a segment of DNA. Gene expression can also be controlled by other genes. So there could be a nearby gene that's actually controlling the expression of this particular gene. I want to make sure that we go back and recognize Mendel, so considered the father of genetics, basically the first person to figure out that something has to be passed on or transmitted from one generation to the next. And then hopefully you've heard of the Human Genome Project, basically sequenced an entire human genome. So now we're kind of going back and trying to find out what each sequence actually means, right? So we know what the sequence is of the entire human genome. We're just trying to find out, okay, well, these right here, this makes protein X, right? Some of these slides are going to be kind of a recap for us because we covered some of this material in chapter 27 when we did reproduction. So hopefully we know the diploid number for a human is 46. So that's the total number of chromosomes. We have 23 pairs of homologous chromosomes. So if I have this one, and these are both chromosome, let's say number 12, right? I know they don't look exactly the same. I'm a terrible artist. All right, chromosome 12. So if this is your cell, so this is you have 2 copies of each chromosome. That's why there are 23 pairs. This one you got from mom, and this one you got from your dad. We also have 1 pair of sex chromosomes. The other ones, they'll call them autosomes. So we have 23 pairs of homologous chromosomes. One of those 23 pairs is a set of sex chromosomes, and the rest of them we use this term autosome. I opened the chapter with a karyotype. We got to see all of the diploid chromosomes displayed in their homologous pairs. Genome, this is the genetic makeup. So all the genetic instructions that make you who you are. Alleles are matched genes at the same locus on homologous chromosomes. We already saw this. So here's, I can't seem to make that second one the same size, right? All right, so if these are both chromosome 12, and right here I have genes that code for hair color. I can have one allele that says brown hair, and I can have another allele that says blonde hair. If they match, if it's brown and brown, if I have 2 browns, we say this is homologous. The alleles controlling a single trait are the same. So what's heterozygous? What if I had one brown allele and then I have one blonde allele? That would be a heterozygous. So what does it mean if I say you have a dominant allele? A dominant allele is going to mask or suppress its recessive partner. So again, if I have chromosome, so this is, we'll keep doing chromosome 12, right? And here I have alleles, right? Genes, alleles for hair color. Brown is dominant over blonde. So brown would be the dominant allele. So if you had one brown allele and you had one blonde allele, the blonde is masked. So what's your hair color? What does it look like physically? You have brown hair. So there's a lot of dominant and recessive partners. Probably one recessive we're familiar with is red hair, right? So red hair, the only time red hair shows up is if you have two recessive red hair alleles. This is why you could go through many generations and then all of a sudden a redheaded child pops up, right? Maybe great-great-great-great-grandmother was a redhead. That allele stayed hidden through all of those generations, and then eventually that red allele became exposed or became physical because the mom and the dad both have the recessive red-haired allele, and that happened to be the combination of egg and sperm. Another way that we can talk about genes are genotype versus phenotype. Genotype is genetic. So I like to think, what's on the chromosome? So whatever's on the chromosome, if you have a brown allele and you have a blonde allele, right, that's your genotype. So they usually do, the little b would be recessive and the capital B would be the dominant. Phenotype is what you actually look like. What's expressed? What can we physically see? So again, could I have one brown allele and one blonde allele genetically on my chromosome? Yes. But what do I end up looking like? I have brown hair because the brown is dominant over the blonde and masks the blonde. So if you just see somebody with brown hair, do you know that they have this hidden blonde allele? You probably don't, right? So phenotype is what do I look like? Genotype is what does my DNA say? What genes are on my chromosomes? Here I have listed the three sources of genetic variation. So we're going to talk about independent assortment of chromosomes. We're going to talk about crossing over of homologs, homologous pairs, and then we're going to talk about the random fertilization. And all of these introduce genetic variation into our offspring. First thing we're going to talk about is segregation and independent assortment. So when developing gametes, gametogenesis, the maternal and the paternal chromosomes are randomly distributed into the daughter cells. So we're talking about when you go from this 46 to this 46 and this 46, we're saying these chromosomes that were here that copy themselves, those 96, were randomly distributed. Two alleles of each pair are segregated during meiosis 1. Alleles on different pairs are distributed independently. We don't line them up. We don't say, okay, all maternal on this side, all paternal on the right side, all maternal on the left side. Okay, everybody line up and we'll split down the middle. They could be a random assortment. This is a nice illustration of that effect. So let me get my little colors going here. So here's your original cell. Okay, so this is you. Why did we color code them? This is the chromosome you got from your mother, and this is the chromosome you got from your dad. Now I'm going to take your cell and it's going to undergo meiosis. What do I do with this cell? I copy the chromosomes, right? We started with 46. I'm going to copy all the chromosomes and get, oops, I lied, 92. So what happened to these? What happens to this chromosome? Copies itself, right? So now I have 2 of them. What happens to the chromosome that originally you got from your dad? Well, it copies itself. So what's happening here? They're all lining up. This is meiosis 1, metaphase 1. You're making gametes. The whole point is making gametes, right? So this is your cell making gametes, but we're looking at these color codes. They're based on your genetic information that you got from your parents. So what they're trying to show you is the lineup of your copied chromosomes are random. They're independent. They don't line up. There's no parking space. It's not like chromosome 1, chromosome 2, chromosome 3. They can be in any order. And your maternal can be on one side and your paternal on the other. And then here on this side, we've got paternal on one and maternal on the other. Well, remember what's going to happen. These are all going to split and come off. And then they're going to split again and you're going to get gametes. So during meiosis 1, metaphase 1, the homologous pairs of chromosomes line up independently. Therefore, they're going to end up in different daughter cells. In segregation and independent assortment, because of meiosis 1, metaphase 1, the way that the chromosomes line up is independent, we can get 8.5 million different combinations. So when we go from that 46 and then all the DNA copies itself, we end up with 92. What we're saying is when we get to this stage, we have millions of different combinations of chromosomes. You could have chromosome 1 is mom's, chromosome 2, dad's, right? Goes back and forth. And what are we doing? We're making gametes. So you're passing on genes from your parents to your offspring. And we have about 8.5 million different combinations because they do not line up in a specific order during metaphase 1. The second event that happens is the crossover in genetic recombination. So remember that we have those homologous pairs and that they can cross over and exchange segments. So here are your original chromosomes, right? So this is original, this is you, right? This is your cell. And what are we trying to do to this cell? We're trying to make gametes, right? So this was originally the chromosome you got from mom, and this was originally the chromosome you got from dad. But this is you, right? This right here would be you. That's our first 46 that we start with. What do we do to every chromosome before we enter meiosis? We copy everything. So here, your mom has recessive hair color and it's blonde. So recessive blonde hair. And mom has blue eyes. Dad, these were your parents. Your dad had brown hair allele and a brown-eyed allele. What did you have? This is you. You're carrying recessive alleles, but the dominant ones take over. So you've got brown hair and brown eyes, just like your dad. But you have these recessive alleles hiding. So what do you think happens if we cross these alleles? You're going to end up with variation. So here's the crossover event where we're going to swap these two alleles. So we've now officially swapped sections, right? So remember what happens. We're here at this point. We've got 92. That's where we're at right now. What happens to those 92? They get split into 46 and 46. So right down the middle, these two chromosomes are going to go to one cell, and these two chromosomes are going to go to another cell. So now what happens? This is gamete 1, 2, and 3, right? So we had them together, we split down the middle, and then each one got split. So we went from 46, 46, and then we split to our 20, oops, that's supposed to be a 3, 23, 23, 23. So this chromosome is going to go to gamete 1, this chromosome gamete 2, this one 3, this one 4. Do you see now that we have more variation than what we started with? What happens if this gamete becomes the ovum? What are we bringing with it? Recessive blue, recessive blonde. What if this one becomes the ovum? Now we've got blue-eyed gene and a brown-haired gene. The last thing that introduces genetic diversity is random fertilization. So since we don't know which sperm or which egg are going to unite and form the zygote, that also adds more diversity. So basically, an offspring represents 1 out of 72 trillion possibilities. So because of independent assortment, because of random fertilization, and because of crossing over, we have trillions of possibilities of possible offspring. We need to go through and talk about patterns of inheritance. Something to keep in mind is we're going to keep this very simple. We're going to say there's one brown-haired allele and there's one blonde-haired allele. But there are several gene pairs sometimes that have to interact in order to produce a phenotype. So eye color, we try to make it really simple in the textbook, but it is much more complicated. So eye color, there are 3 alleles on different chromosomes that we found so far that can control eye color. First is dominant and recessive. So we've kind of seen an example of that already. If we had the big B, that meant brown hair. And if we had the little b, meaning recessive, we have blonde hair. Then they give you this example: dominant allele, big T tongue roller, recessive allele, little t, you cannot roll your tongue. So if you're big T, little t, you're homozygous for that allele. If you're big T, little t, you're heterozygous for that allele. So let's go to the next slide and look at how we came up with these different genotype and phenotype ratios. Basically, we're doing what's our best guess, right? What are the chances that your offspring is going to be a tongue roller versus the chances that your offspring cannot roll their tongue? So here's the Punnett square. This female is hetero, I think it's, there we go, hetero for the 2 types. So she has 1 allele for tongue rolling and 1 not. He has 1 allele for tongue rolling and she has 1, or sorry, he has 1 for not. So how do you know which allele is going to end up? Remember, we have 2 of every chromosome, and we're going to have that during meiosis. We're going to go from diploid to haploid. So is it just as likely that mom could give the big T as it is mom could give the little t to the ovum? Yes, it should be. You have a 50-50 chance, right? What about the male? Do you have an even chance that he'll give the big T versus the little t? Which one is going to be in the sperm? Right? We don't know. So it's 50-50. So what we do is we put the big T on this side, little t on this side for mom. That's what mom can give. Dad can give big T or little t. These go this way. These go this way. So if the big T and the big T meet each other, if this is the sperm and this is the egg, you're going to have an offspring that's big T, little t. If this happens to be the sperm that gets into the ovum, and this is the chromosome that's in the ovum, you're going to get this heterozygote, just like mom and dad. And you can see this is little t, big T, little t, little t. So what they're asking is, or what they're trying to show you being shaded, is how many can roll their tongue. What do you need to roll your tongue? You just need a big T. How many of them have a big T? 3 out of 4. So it's probable that about 75% of your offspring will be able to roll their tongue. What about this one? The only way you could have a non-tongue roller, right, is if you have 2 recessive alleles. So they have a 1 in 4 or a 25% chance of having a child that cannot roll their tongue. So I know that this seems silly. We're talking about tongue rolling, who cares? But you could apply this to brown hair versus blonde hair, right? So you could have 3 children that are dominant brown-haired allele, and then you could have 1 child that has blonde hair because it got both of the recessive alleles. There's a whole list of dominant and recessive traits. So I think there's an image coming up next, figure from your textbook. They just want to mention that dominant disorders are usually uncommon because they become lethal and result in death before that individual can reach reproductive age. Most of our genetic disorders are caused by recessive traits. So when you have 2 recessive alleles, that usually causes the disorder. And why do we never eradicate it? Well, because we always have these heterozygotes secretly carrying this recessive trait, even if it's bad. And then if they happen to meet another individual that has the recessive trait, there's a 25% chance that they're going to end up with a child that has the disease. Incomplete dominance is when individuals have intermediate phenotypes. So there's a simple one, let's say you could be red, right? You could have, let's say, pigment in a flower. You could have a red flower, and you could have a white flower. The white is recessive, the red is dominant, but then you have this intermediate flower that has pink pigment or pink petals. So if your hetero has its own phenotype, that would fall under this incomplete dominance. We've already seen one here with sickle cell. So if you have normal sickle cell, you have 2 big S's. If you carry the sickle cell trait, you have 1 big S and 1 little s, right? You're heterozygous and you're carrying the recessive trait. If you have 2 recessives, you have sickle cell anemia. We've seen multiple allele inheritance, basically if the genes exhibit more than 2 allele forms. So what we're saying is on your chromosome, can you have an A allele? Yes. Could you have a B allele? Yes. Could you have an O allele? Yes. So if there's more than 2 allele forms, then it's going to be multiple allele inheritance. We also have sex-linked inheritance. We talked a little bit about this when we talked about hemophilia. So there are some traits that are inherited on sex chromosomes. Mostly X chromosomes, they have several genes. The Y chromosomes have fewer genes. Sometimes we refer to these as X-linked genes because they're found only on the X chromosome and they're passed from mothers to sons. Remember, if you're a mom, right, you have XX. If you're a dad, you have Y and X. And then if you're mom, all you can give to your son is X because dad has to provide the Y, right? So if mom has something defective, right, if mom has a defective gene on her X, her sons are going to see the full force of it. If mom had a daughter, this X was from her dad, and this X is from her mom. If mom's X was deficient, dad's X could take over because you've got a backup copy. Well, males don't have a backup copy, right? We also have this term polygene inheritance, poly meaning many, right? So this depends on several gene pairs at different locations all acting together or in tandem. Skin color is a good example. Eye color, I told you there's 3 different chromosomes with different alleles that all impact eye color. Height is one of those, and skin color. So let's look at skin color as an example. If you have really dark skin, you have multiple dominant alleles. If you have light skin, you're going to have multiple recessive alleles. So think of all the possible combinations. So here we have a very light skinned parent, and they're mating with a very dark skinned parent, and we end up with these heterozygote first generation offspring. So next we're going to look at what's the second generation. If I took two heterozygous parents and I mate them, what are their offspring chances or percentages, right? So I could donate this allele or this allele. I could donate this allele or this allele, this allele, this allele. Boy, we could go back and forth on this, right? Try doing a Punnett square with 3 different genes. It's hard. So I just want you to get the basic idea that if I take 2 heterozygous parents and I mate them, I get this nice kind of peak, right, proportion of the population. What's the chance that I get this little a and this little a and this little b and this little b and this little c and this little c? What's the chance that I'm fair-skinned? It's pretty low. What's the chance that I'm extremely dark-skinned? Very low. Where do we get the most probability? Right in the middle, right where we're heterozygous. So this is how skin color is determined. It depends on how many dominant alleles you have. So if you increase the number of dominant alleles, what happens to your skin color? Oops, darker skin. So there's a direct relation between dominant alleles and recessive alleles and the number that you have with these polygenic inheritance, like skin color and height. I want to make sure that we understand that environmental factors can influence genetic expression after birth. What we're saying is, let's say you have genes to be tall, right? You have dominant alleles and they all say you're going to be 6'5". What happens if you don't have enough food? Right? If you have low food consumption, maybe you have low ATP, right? You are able to do, right? You diminished work, right? Less work in the body. What happens if you have hormonal deficiencies? What happens if you have decreased growth hormone? What happens if you have decreased T3 and T4? That's going to affect your skeletal growth. So environment can affect your genetic expression, whether a gene is on or off, or whether it's on high enough or low enough. There are a few non-traditional inheritance. I just want to talk about this. There's something called small RNAs, epigenetic markers, and then I really want to focus on this mitochondrial inheritance. These small RNAs are really interesting. I'm not going to test you on it, but I want you to at least have heard of them. They're microRNAs, or sometimes called interfering RNAs. These are RNAs that can affect DNA directly, right? So they can inactivate genes on DNA. They can control the timing of events, right? We're thinking that these interferon, if we can figure out how interferons affect your DNA, would it be possible to create drugs or treatment where we could turn on a section of DNA that happens to be off? So let's say you have a tumor suppressor gene, and that gene is on your DNA, and for some reason your tumor suppressor gene has been turned off. Could we create an RNA that could go into your DNA and turn this tumor suppressor gene back on? So that's kind of what we're looking at. So there's something called epigenetic markers. I just want you to realize that we have DNA, right? It copies itself into RNA, and then it translates into a protein. Sometimes these proteins can come back and silence sections of DNA. So it's proteins that we make that can bind to DNA, and they can tell either transcription, copy, on, right? Or they can say silence. So something else to consider, most of us don't think about this unless you've had a biology course, we have DNA outside of our nucleus. So your mitochondria has its own genes, it has its own DNA. It's mitochondrial DNA, and this is transmitted by the mother in the cytoplasm of the egg. So we can test lineage from mother to daughter to granddaughter or grandson based on the mitochondrial DNA. So your mother's mitochondrial DNA is the same as yours, and your child's DNA is the same in the mitochondria as yours. So there is no variation here, or very little variation, but there is no recombination, there's no crossover, there's no nothing. So we can look at the if we had two samples, we had sample 1 and we had sample 2, we could tell if they were related by looking at mitochondrial DNA. Genetic screening. Newborn infants are routinely screened for a number of genetic disorders. There's some listed here. If you've never seen it, they have like these little heel stick cards is what I call them. So when the baby's born, they prick their heel and they put little drops of blood on the card, and then each section goes to a different department and they test it for a number of genetic disorders. One that's easily testable is sickle cell. So and I think every state tests for sickle cell disease. If we find out that you have sickle cell day 1 or 2, we can create a medical plan for you, and that will increase your lifespan. PKU, you cannot break down one of the amino acids, which is phenylalanine. So if we know from day 2 of your birth that you cannot break down phenylalanine, we can try to avoid any food that has phenylalanine in it. So yes, that doesn't sound like a lot of fun, but it's better than having phenylalanine build up in your child's system and start causing mental delay issues, right? So there are other avenues that you can use to identify carriers. You can look at pedigrees. So basically it's a family tree, right, of all of your relatives who has which disorder if you happen to know it. And this will help to predict the future. We can also do blood tests and DNA. So that karyotype that we saw at the very beginning, we could look at your DNA and we could probe it for a very specific disorder. And that way we would know if you had Tay-Sachs or cystic fibrosis genes, we can identify them right away. And some people want to have these particular disorders tested for before they decide if they're going to have children or not. If you're both a carrier of Tay-Sachs, right? If you both carry the recessive allele, you have a 25% chance of having a child that has Tay-Sachs disease. Here's the pedigree. You can see they'll have boxes or circles, and that will tell you male or female. And then you can see who's been affected and who hasn't been affected. So this one is if you're affected, you're blue. So we can see in this case, it's the heterozygous combination that creates the widow's peak. So if you're homozygous, you have no widow's peak. If you're heterozygous, you have the widow's peak. We know sometimes that fetal testing is necessary. So if you have a known risk of a genetic disorder, we can do an amniocentesis or we can do chorionic villi sampling. So an amniocentesis is when we pull out the amniotic fluid, and there are fetal cells in the amniotic fluid. And then we can test those fetal cells. They're going to have DNA, right? And we can look for genetic abnormalities. The second is the chorionic villi sampling. So remember, chorionic villi were made from fetal membrane. There we go. So this is going to have, again, fetal DNA. So we can pull a sample of chorionic villi and we can karyotype it. This one's a little bit more risky because you're actually removing, remember the chorionic villi are laying in the pools of mom's blood. So this is a little bit more risky. Neither one of these look like a lot of fun, right? Here's a syringe. We're putting it through the abdominal cavity of the female. We're pulling out amniotic fluid. We're collecting the fetal cells and we're growing them up and we're doing a karyotype. Or we can come in and remove some of the chorionic villi and we can take those fetal cells and also do a karyotype. There are some time limits here. This is about 8 to 10 weeks, and this is a little bit later, 14 to 16 weeks. Last thing to look at is human gene therapy. I just want you to realize that we are trying to cure some of these disorders, something like sickle cell, right? What if we could go in and just fix that one protein that's in the wrong spot and create a fully functioning new protein? What if we could go in and inject the correct gene directly into the gene that's not working properly? So you might have heard of CRISPR. CRISPR is genetic technology that allows us to insert pretty precisely into the genome. Does that mean that's something we really want to start doing tomorrow? No, we probably need lots and lots of research on other animals before we ever move into humans, right? But imagine if you could completely pull out a defective gene out of your genome and insert the proper gene into your genome, and then you never have that particular disorder. So there would probably be lots of disorders that we could cure if we can get this injecting a corrected gene into cells, if we could get this down to where we're not making any mistakes. Again, you have to be careful, because what if you insert that gene in the wrong place? Just one letter of DNA different, and you could be disrupting another protein from being made, or you could turn a gene off. So we'd fix one problem and then we'd create another problem. So we have to kind of be careful there.